On behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia series and our sponsor, Biodesix, I'd like to welcome you to SARS-CoV-2 Neutralizing Antibody Testing for Vaccine Efficacy Assessment. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenters for today. First is Sean Taylor, MBA, Ph.D., Field Application Scientist Manager, North America, GenScript. Our second presenter is Laura Peak, Ph.D., Vice President, Laboratory Operations, DeSoto, Kansas, Biodesix. With that, I will turn the presenter ball over to Dr. Taylor, and we'll bring his presentation up now.
Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And here I'll be talking about a novel SARS-CoV-2 Neutralizing Antibody Test for vaccine efficacy assessment. So with the initiation of the global SARS-CoV-2 vaccination programs, some critical public health questions come to mind. The first one is associated with the widely different efficacies of the vaccines themselves and how protection is assured post-vaccination, especially in the context of the wild type and circulating variants of the strain. Furthermore, how long does immunity persist after the administration of vaccine? And the third question is associated with the testing. So what tests now can give us the most actionable information in populations of mixed vaccinated and non-vaccinated individuals? So the major tests for SARS-CoV-2 fall into two major categories: serology versus molecular. And in the early days of the pandemic, the predominant test was molecular testing because that permitted the ability to assess the level of virus circulating in an individual by measuring the RNA from the virus. So the RNA was measured by a molecular test called PCR. And if you see in this graph here, from the onset of symptoms, the RNA can be measured very early in time from the onset of symptoms. And this was important to delineate between infected and non-infected individuals so that they could go into quarantine and prevent the spread of the pandemic. The immune response antibodies typically persist for a long period of time, months and possibly years. We don't know this information yet for SARS-CoV-2, but for sure they persist much longer than the viral RNA. And that's the big difference between a molecular test and a serology test. So for a molecular test, you have a very narrow window of opportunity very early in the onset of symptoms from the virus to measure the RNA, because eventually the immune system will take over, clear the virus, and then all we'll be able to measure are the immune response antibodies. Now, conversely, a serology test should never be measured early, because early on, after symptom onset, there won't be an immune response generated yet to measure those antibodies. So you could get a negative serology test if you measure too early, and you can definitely get a negative molecular test, PCR test, if you measure too late post-symptom onset. SARS-CoV-2 will replicate itself by binding to the ACE2 receptor, as we see here in purple, of the epithelial cells that line the lung. So it does this via the spike protein, and in particular the receptor binding domain of the spike protein, which is the part that interacts with the ACE2 receptor, as we see here. And when that binding event happens, then the RNA from the virus will be injected into the cells, and the virus can then use the cell machinery to replicate itself and propagate and spread the infection within an individual. So the goal for our immune response antibodies would be to stop this binding event. If this binding event can be blocked, then the virus would be rendered inert, unable to replicate itself. Of all the antibodies that are generated in an immune response, there's a subset of special functional antibodies that are called neutralizing antibodies that can bind to the spike protein of the SARS-CoV-2 virus in such a way that the virus can no longer interact with the ACE2 receptor. And when this happens, the virus is considered blocked or neutralized. And that's why we call these neutralizing antibodies. So these are the antibodies that we would like to have either from an immune response to natural infection of the virus or from vaccination. So let's use a dartboard analogy to represent the immune response to SARS-CoV-2 infection. So the dartboard can represent the virus. This is SARS-CoV-2. The bullseye of the dartboard is the specific antibodies that we want to generate that will bind to the receptor binding domain of the spike protein around the virus to prevent it from binding to the ACE2 receptor. So we want very specific antibodies that are generated right here at the bullseye of the dartboard. Now, the immune system doesn't know that it needs to specifically generate antibodies to the receptor binding domain. All it sees is the virus as an invading particle, and it's going to generate antibodies to all different parts of the virus and all different parts of the spike protein as well. A small number of those antibodies will be to the receptor binding domain. So here you see all these different antibodies that were generated, and none of them are very well even binding to the dartboard or the virus. And over here we have another depiction where we now have some antibodies that were generated that bind to the virus in various places. And only one, the green arrow, has made it to the bullseye. So we have one antibody generated in this depiction that will bind in such a way that it will block the ability of the virus to bind to the ACE2 receptor. Now, fortunately, the receptor binding domain of the ACE2 receptor is quite highly immunogenic. So you will get many darts that hit the bullseye, but they will still not represent the majority of the antibodies. The majority of antibodies will likely bind to various parts of the virus, and you'll get a subset of very specific antibodies that are neutralizing that will bind to the bullseye. There are two currently available tests that are considered the gold and silver standard virus neutralization tests. And those are this VNT, virus neutralization test, and the pseudovirus neutralization test, abbreviated PVNT. The way these tests work is by incubating either the live SARS-CoV-2 virus, in the case of the VNT test, or a lentivirus that's expressing just the spike protein of the SARS-CoV-2 virus with an individual sample. And if that person generated neutralizing antibodies to the virus, then the virus can no longer interact with the ACE2 receptor of live cells that are also in this assay. In the case of a sample that does not have neutralizing antibodies, as we see here, the virus can strongly bind to the ACE2 receptor, and that generates some kind of signal, either a luminescence signal, or in the case of a plaque reducing test, we would be able to count the difference in the number of plaques between the binding event or a non-binding event. Both of these tests are very good, very elegant, but unfortunately, they require 2 to 4 days of time just to generate the data and very complex equipment and reagents in both cases. Furthermore, for the neutralization tests, since it uses the live SARS-CoV-2 virus, this is a dangerous test that further requires a BSL-3 containment facility. So even more arduous to perform this kind of test. So in both cases, these two tests, although they're very good, they really are not amenable to high throughput testing. So GenScript partnered with Dr. Linfa Wang to create a test that teases out the protein components that interact in the virus ACE2 receptor by purifying just the receptor binding domain of the spike protein, which is depicted here in brown, And the extracellular domain of the ACE2 receptor, which is depicted here in green, we were able to generate the same strong interaction just between these protein components. So now we've eliminated the need to use live virus and live cells in an assay by plating the ACE2 receptor on ELISA plates and then interrogating the interaction of the receptor binding domain to the ACE2 receptor in the presence of antibodies from individual samples. So the way this test works is exactly the same as the live virus test works, except the incubation of the patient sample is simply with the protein, the receptor binding domain. And if the individual has neutralizing antibodies, which are depicted here in red, then the receptor binding domain can't bind to the ACE2 receptor. And when we wash the plate, these complexes will be washed away, and we will ultimately generate a very low or even no signal in this well. Conversely, if we test an individual who doesn't generate neutralizing antibodies, remember that dartboard, we can have lots of darts going to different parts of even the receptor binding domain that will not prevent it from binding to the ACE2 receptor. So these would be non-neutralizing antibodies that are not functionally blocking the interaction. So therefore, the receptor binding domain can bind to the ACE2 receptor. And because we've conjugated in blue here horseradish peroxidase to the receptor binding domain, if this binding event occurs, when the plate is washed, this is a very stable complex. So the RBD will remain bound to the ACE2 receptor after washing. And when we add TMB, it will cause the horseradish peroxidase bound to RBD will cause the solution to turn yellow. So we'll generate a signal. So the more yellow the color, the more intense the signal in terms of a yellow color, the less neutralizing antibodies are in that sample, down to almost a clear color if the individual has a lot of neutralizing antibodies. So a very elegant, simple test invented by Dr. Lin-Fa Wang and commercialized by GenScript. So what are the advantages of the cPass test? The first major advantage is that it's immunoglobulin agnostic. So remember, our bait protein, which is RBD, we're using RBD in the sample to bind to the neutralizing antibodies. It doesn't depend on a secondary antibody for IgG, IgM, or IgA. Any subclass of immunoglobulins can bind to the receptor binding domain. And if that subclass of immunoglobulins generates neutralizing antibodies, a signal will either not be generated or generated. So this is a very nice test to assess the total neutralizing antibody response from all the classes of immunoglobulins that would be generated against either vaccination or infection, which dramatically increases the sensitivity of this assay versus a more traditional serology test that just looks at binding antibodies to coded antigens on the surface of the plate. Which brings me to the next advantage, that the RBD is in solution. It's not coated on the plate. Remember, the RBD is in solution, so the RBD is in a near native conformation, which forces the antibodies to bind to the epitopes that they would otherwise find in an in vivo situation. So these antigens that are exposed on the RBD are in a near native conformation, dramatically increasing the specificity of this assay because the antibodies need to very specifically bind to those native epitopes. In a more traditional binding assay for IgG or IgM, a traditional serology test where the RBD would be coated on the plate, that can alter the conformation of the spike protein or RBD, or even in nucleocapsid protein tests that are coated to the surface of plates or coated on beads. It can change the conformation of the protein, and lead to the exposure of antigenic sites that would be non-native and potentially non-specifically bind other immune response antibodies that are unrelated to SARS-CoV-2, leading to false positives, decreasing the specificity of those assays. So this confirmation of assay increases specificity. And finally, the third advantage is that this is a dual detection and functional assay. Remember, we're detecting the interaction between RBD and the ACE2 receptor by the interaction of antibodies to RBD. So this is a functional test. This is not just a binding test where we're looking at antibodies that just bind to RBD. We're looking at the specific antibodies that block the interaction of RBD to the ACE2 receptor. So we're specifically detecting those bullseye arrows that I showed you in that previous dartboard slide that are neutralizing. So we have done several experiments to verify the specificity of this test against traditional ELISA binding antibody tests. And here's one study. This was published recently in the Journal of Clinical Microbiology, where we looked at commercially available RBD-coated ELISA plates, spike-coated ELISA plates, and nucleocapsid-coated ELISA plates. And we looked at the same set of 68 samples, which contained 45 presumed positive. These were PCR positive samples, and 23 presumed negative, but these were pre-pandemic, so they really should be negative. These samples were acquired before the pandemic occurred. And if we look at on the right side of all of these graphs, these are the negative samples. And we can see that in the RBD-coated plates, there were 3 samples that were above the cutoff, which were deemed falsely positive. So this is unfortunate, especially for pre-pandemic samples that should really be negative. Same thing for the nucleocapsid-coated plate. One of those 23 samples was called falsely positive above the cutoff for that nucleocapsid-coated plate. So again, very unfortunate, especially when the samples are truly negative and pre-pandemic. The Spike S1 coated plate did call all these samples negative in this particular experiment, and same thing with the cPass neutralizing antibody test from GenScript. So this test called all samples negative. For the positive samples on the left side in circles, there were 45 presumed positive. Now, although the RBD called 43 of the 45 positive, it's very difficult to assess the validity of these data because of the number of false positives that were called. For the Spike S1 coated plate, which did call all the negative samples properly negative, there were still quite a few false negative samples for the positive samples. Only 34 of the 45 samples were called positive, 11 were called falsely negative. And for the nucleocapsid coated plate, only 28 of the 45 samples were called positive. For the cPass test, 39 were called positive, 6 were called falsely negative. We were a little curious about these 6 samples. Unfortunately, we couldn't get any of the PCR data, the CT values for these results to know if perhaps these samples may have been called falsely positive. But we did correlate these 6 samples with the other 2 tests that had less problems with specificity. And we found all the same 6 samples were called negative in both the nucleocapsid test and in the spike test. These are the circles in red. So the same 6 circles in red were negative for nucleocapsid and also for spike. So it just brings into question whether or not these 6 samples were in fact either didn't seroconvert or were truly just negative samples that were called falsely positive by qPCR. So we also did another part of this study with Health Canada. So the same article, just a different study, where we looked at samples that were delineated by the number of days post-symptom onset of COVID. So all of these were natural infection and recovery from COVID. And if we could look at the samples that were 5 to 9 days, we see that the sensitivity of the cPass test, so the GenScript cPass test, was very comparable to the sensitivity of the other tests. The diasorin and Euroimmune tests actually didn't do as well in the early days post-symptom onset. In the 10 to 19 day period, actually, the sensitivity of the GenScript test, of our neutralizing antibody test, was actually the best. So it gave the highest sensitivity within 10 to 19 days of sampling post-symptom onset. And at greater than 19 days post-symptom onset, all the tests did equally well, 100% sensitivity for those samples. If we look at all the time points together, the sensitivity of the GenScript test was in fact at the top of the heap at 93% versus the other tests that were used in this study. The only other test that gave the same level of sensitivity was the affinity test. We also looked at the specificity of the test by looking at pre-pandemic negative samples, approximately 50. And you can see the GenScript test called all those samples negative, so 100% specificity. There were some tests that falsely called some of those samples positive, unfortunately. So if you look at the positive and negative predictive value and the accuracy of those tests, GenScript fared very well, again, at the top of the heap of all those tests. And we attribute these results to, again, the way in which this test is made with the RBD in solution, and the RBD able to detect all immunoclasses of immunoglobulins that are neutralizing. So we also compared cPass to the gold standard live cell assays. With 66 samples, cPass was able to delineate 40 negative, 26 positive, versus plaque-reducing neutralizing tests. So this is an actual VNT test that uses live SARS-CoV-2 virus and live cells. cPass called 26 of the 66 samples positive, 40 negative, and with PRNT at a higher stringency, which is what's recommended to be used with this type of test, either PRNT 75 or 90, 39 of the samples were called negative and 27 positive. So only one sample in red here was called negative for cPass, but positive for PRNT. So very close correlation. You can see that when the analysis stringency drops to a 50% plaque reduction in the live cell test, 2 more samples become falsely positive. And this is an inherent problem with these live cell tests and the analysis stringencies that are set that can change the data from one analysis stringency to another. If we move to comparing cPass with Foci-Reducing neutralizing test, and this is, again, it's a very similar test to the plaque-reducing test using live cells, we see that for 45 presumed positive samples, the data changed dramatically between analysis stringencies in the Foci-Reducing test. When we go from 50% to 90% analysis stringency, we go from 44 positive samples all the way down to only 23 positive samples at 90%. So the data changes dramatically between these stringencies, which again begs the question, how do we calibrate these live cell assays to assure that the data truly reflects the samples in terms of positive and negative? The cPass test was very consistent, calling 6 samples negative and 39 of those 45 positive, which compared closely to a FRNT 75 analysis stringency. So perhaps cPass could be used as a calibrator to help calibrate these kinds of tests to get the correct results that reflect the samples. Several other research groups have looked at using cPass as an alternative to the gold standard live cell tests. And this is one publication where they looked at 401 human samples, some dog, cat, and hamster samples, because again, cPass is species independent. It can be used in any kind of animal model or even with cells. And the correlation of cPass neutralizing test between the plaque-reducing test at a 90% stringency for this study was excellent. 99% sensitivity and 99% specificity. We have produced a protocol for the semi-quantitative analysis of neutralizing antibodies using cPass. And by running samples, unknown samples, against the standard curve, we can generate the titer of neutralizing antibodies in each of those samples. And by multiplying the titer from the standard curve by the dilution factor used for each of those samples to be within the linear range, so each sample needs to be diluted to be within a linear quantitative range before interpolation with the standard curve. By multiplying those titers by the dilution factor, we get a final neutralizing antibody titer within each sample, which then translates to a fold difference. Applying this semi-quantitative approach to 25 samples from individuals who were infected and recovered from SARS-CoV-2 with sampling taken approximately 3 to 4 weeks post-symptom onset, we were able to assess the difference in the final neutralizing antibody titers from one individual to the next. And you can see we ranked these in order of neutralizing antibody titer, and the differences were quite astonishing. It was translated to a 610-fold difference between the lowest sample and the highest sample in the amount of neutralizing antibodies that were generated. And this simply begs the question, what is the titer of neutralizing antibodies that might be required to block the virus from binding to cells if infection reoccurs? Are these individuals less likely to block the virus versus these individuals that have a much higher level of neutralizing antibodies? And then the other question, of course, is over time, how long will these titers persist to assure that there's an appropriate amount of neutralizing antibodies to prevent the virus from binding to cells and propagating infection if they ever do encounter the virus? So let's switch gears to now the vaccine era that we're in, where we have mixed populations of vaccinated and non-vaccinated individuals. And let's just look at the vaccine efficacy data from the manufacturers. And this was published data in Microbes and Infection recently by Hotez et al. And I really don't want to go through all of this table. I just want to direct you to a couple of bits of data from this table. Let's look at the efficacy versus 2 doses. So the normal number of doses that are needed to confer full protection with the vaccines. And with the Pfizer vaccine, after 2 doses, it's 95% effective. But if we move down the vaccine ladder to the Astra vaccine, it goes down to 70 and even 62%. And if we go further down to the Sinovac vaccine, it's only 50%. So there's a wide range in just efficacies of these vaccines. Now, if we move to efficacy versus the different strains of virus, what we see here is that if we look at the J&J vaccine, its efficacy goes from 72% down to 57% in the presence of the South African strain of COVID-19. It's even worse for the AstraZeneca vaccine. And when we look at AstraZeneca, we're down to only 10% efficacy in the presence of the South African strain. And this is why the AstraZeneca vaccine is actually no longer used in South Africa. So there's a big concern within the general population simply about the efficacy of the vaccine. Is the vaccine that I've taken giving me adequate protection from infection to SARS-CoV-2 and the associated variants? In a very recent study that hasn't even been officially published yet, it's only on the preprint server, but very nice data in this article, they were looking particularly at elderly individuals just with the Pfizer vaccine. And what they found in this article was that 30% of people that are over the age of 80 do not even elicit a measurable level of neutralizing antibodies after both doses of the vaccine. So, over the past year and a half, there's been a rapidly increasing body of evidence that supports the correlation between the presence of SARS-CoV-2 neutralizing antibodies and inhibition of viral propagation. This has also already been shown for other viruses, so this is not a big surprise. The big concern is associated with vaccines and the efficacy of vaccines, where we've already shown from the previous slide that there's a marked decrease in efficacy, particularly with the South African variant, and in particular with the AstraZeneca vaccine. However, these prevailing data from a variety of articles shows that for many of the vaccines, the neutralizing antibodies, the power of those antibodies is still quite robust in neutralizing the variant strains from binding to host cells. So although there might be some vaccines that are much less effective, most vaccines are more than adequate to generate the level of neutralizing antibodies that are required to prevent the virus from binding to host cells and propagating. So all of this leads to a general conclusion, therefore, that the direct measurement of neutralizing antibodies that are elicited post-vaccination could therefore be a strong indicator of a productive and a functional response to SARS-CoV-2. And this can be measured now in a simple cPass test. So in much of the data that's been published using samples from people who were naturally infected by SARS-CoV-2 and recovered, we found that by and large, the neutralizing antibody levels correlate very well with the binding antibody levels. The data are either a little better or similar between the cPass neutralizing antibody test and the commercial IgG binding antibody tests. However, this was not the case for vaccination. And if we look at the first dose of the Pfizer-Moderna vaccine, so this is a study that was recently uploaded on the preprint server, what we see is that in comparing two different IgG binding antibody tests in light blue and medium blue color, the neutralizing antibody test from GenScript was always significantly lower. There was a lower proportion of people who elicited neutralizing antibodies versus almost 100% for Pfizer of binding antibodies, either 2 weeks after the first dose or just before the second dose. It was practically 100% of them had a total binding antibody response, but only a fraction of them had neutralizing antibodies. For elderly people who received the Moderna vaccine, the result was very similar. There was only a proportion of individuals who actually had a neutralizing, detectable levels of neutralizing antibodies, either 2 weeks after the first dose or just before the second dose. So this is interesting data. It definitely contrasts how the neutralizing antibodies are elicited post-vaccination versus after natural infection and recovery to determine whether someone has had a productive neutralizing antibody response to vaccination. So in formulating a potential testing strategy for SARS-CoV-2 in a mixed population of vaccinated and non-vaccinated individuals, the following conclusions can be reached. First, we know that SARS-CoV-2 molecular testing only represents a moment in time. A negative test only means that an individual was negative at the moment that they took the test. Molecular testing does not assure protection from future SARS-CoV-2 infection. Second, the presence of binding antibodies, IgGs, does not mean that functional neutralizing antibodies are present, and we just saw this in the previous slide with the vaccine data. Furthermore, neutralizing antibodies can persist for months and possibly longer, but we're not sure if this will vary greatly between individuals. Certainly, a positive neutralizing antibody test indicates the persistence of frontline functional antibodies that can block the virus from propagating. Given that vaccine efficacy varies widely, and we've already seen this from all the commercial publications from efficacy data, a cPass test can help assure that the expected productive and functional response to vaccination was elicited. Neutralizing antibodies can also be measured longitudinally to assess titers over time, and this is from our modified semi-quantitative approach, not yet FDA authorized, but certainly can be conducted using the research-only use kits. So, for mixed vaccinated and unvaccinated population, perhaps a combined approach that utilizes molecular testing, PCR, for those without immunity, and cPass for vaccinated individuals may be the optimal approach to minimize the risk of a future pandemic.
Thank you, Elizabeth, and thank you, Sean, for providing such a fantastic summary of the cPass technology. I would also like to thank Cambridge Health Tech Institute's COVID Breakout Series for hosting today's webinar. I am Laura Peak, Vice President of Laboratory Operations for Biodesix's DeSoto, Kansas Clinical Laboratory. One of the most exciting aspects of my job here at Biodesix is that we get to evaluate and qualify new assays to bring into our laboratory. We are located just on the western fringe of Kansas City and are a sister lab to our corporate laboratory in Boulder, Colorado. With the Platelia SARS-CoV-2 total antibody and other ELISA tests already being run in our lab, and with Biodesix's commitment to our WorkSafe COVID-19 Testing Program and vaccination protocols in place, evaluation of the GenScript cPass Neutralizing Antibody Test Kit was an easy decision. To briefly introduce Biodesix, we are a data-driven diagnostic solutions company. We have two sides to our business, which are interconnected through our proprietary AI platform. On one side, we have our lung-focused diagnostic testing business, where we work directly with physicians to help solve clinical unmet needs in lung disease. On the other side, we provide diagnostic discovery, development, and testing services for biopharmaceutical companies to stay at the front of the therapeutic and precision medicine innovation curve. We have two high-complexity CLIA-certified clinical laboratories, one in Boulder, Colorado, and a second laboratory in DeSoto, Kansas. Both laboratories are New York State CLEP approved, and the Boulder lab is CAP accredited and ISO certified, while the DeSoto lab is COLA accredited. Biodesix is also Canadian Medical Device Establishment registered, and both labs are approved to run tests with FDA emergency use authorization. Historically, Biodesix has been focused on developing diagnostic solutions across the lung cancer continuum of care. Our Biodesix lung reflex strategy integrates the GenScript and VeriStrat tests to support treatment decisions for patients diagnosed with a lung cancer, providing rapid turnaround of results within 36 hours. Our Notify lung testing strategy consists of the Notify XL2 and Notify CDT tests to evaluate the likelihood of an incidental pulmonary nodule being benign or malignant, respectively. These tests together enable physicians to better triage patients to the most appropriate course of action. We are uniquely focused on rapid turnaround times of test results using the appropriate technology to provide information to help physicians promptly address their clinical questions. At the start of the COVID-19 pandemic, Biodesix immediately recognized that we had the expertise and infrastructure to help in the fight, as droplet digital PCR and ELISA assays were already being run in our laboratories for the GenScript and Notify CDT tests respectively. So with a sense of urgency to help fight the pandemic, we quickly jumped to action. In March 2020, we partnered with Bio-Rad to bring a COVID-19 droplet digital PCR test to the FDA for an emergency use authorization to support molecular testing for COVID-19. After receiving authorization in April 2020, we quickly scaled up operations in our Boulder, Colorado headquarters to meet the growing demand, offering turnaround times of 24 to 48 hours for results. With molecular testing in full swing, in June 2020, we again partnered with Bio-Rad to launch a blood-based immunoassay, the Platelia SARS-CoV-2 Total Antibody Test, that was granted emergency use authorization by the FDA. The Platelia Total Antibody Test measures total IgG, IgM, and IgA antibodies to the nucleocapsid protein of SARS-CoV-2, and results are reported within 24 hours. Our Biodesix WorkSafe COVID-19 Testing Program launched in June of last year, and we have continued to build this program, adding the FDA-authorized Sophia-2 SARS Rapid Antigen Test developed by Quidel Corporation, which can provide results within 15 minutes. And soon we will be adding the FDA-authorized GenScript cPass Neutralization Antibody Detection Test, which Sean has just described, to further enhance our WorkSafe program. Like the Platelia Total Antibody Test, results for the cPass test will be reported within 24 hours. The cPass assay is a great enhancement to the WorkSafe program. A positive cPass test for neutralizing antibodies may indicate a previous infection and/or an immune response to vaccination. Used in combination with the Platelia Total Antibody Test, a positive total antibody test can confirm previous infection with SARS-CoV-2. Since the vaccines induce antibodies to the spike protein, the Platelia Total Antibody Test would not be positive for a vaccinated individual who has never been infected with SARS-CoV-2, since that individual would only generate antibodies to spike protein and would not have antibodies to nucleocapsid. So what does it take to be able to offer an FDA-cleared or approved test in a high-complexity clinical lab here in the US? The Clinical Laboratory Improvement Amendments of 1988, or CLIA regulations, define the requirements that must be met. For a qualitative assay, the clinical laboratory must demonstrate with data collected in its lab that the test's accuracy and precision is comparable to what was established by the manufacturer, and that the reference intervals are appropriate for the lab's intended population. If the laboratory decides to make any modifications to the FDA cleared or approved test, then a complete method validation must be conducted to establish the performance specifications of the modified test. For our performance verification of cPass, accuracy was performed to ensure specimens from donors not previously infected with or vaccinated for COVID-19 were negative, and that specimens from donors previously infected and/or vaccinated were positive for neutralizing antibodies. For precision, we considered performance within a run, between days, between instruments, between operators, and between laboratories. Lastly, we showed that the reference interval was appropriate. We also conducted a specimen stability study to ensure specimen integrity would be maintained during transit, even in extreme temperature conditions. In the near future, we plan to validate the semi-quantitative analysis of the cPass technology, which Sean described earlier, to offer in research and clinical studies with our collaborators. In May last year, we started collecting blood samples, roughly monthly, from donors previously infected with SARS-CoV-2, and we later expanded the study to include donors who have been vaccinated. Our cohort of 74 donors now includes donors vaccinated with all 3 currently authorized vaccines. Blood was drawn into K2 EDTA tubes and tested prospectively with the Platelia Total Antibody Test for anti-nucleocapsid antibodies, and retrospectively with the cPass test for neutralizing antibodies to spike. The ELISA assays were performed in our DeSoto, Kansas laboratory. For those donors who were previously infected, the Bio-Rad SARS-CoV-2 ddPCR tests were performed in our Boulder, Colorado laboratory. The longest follow-up for any donor is currently at 327 days since the first specimen was collected. I will present case studies for a variety of donors to reflect the breadth of immune response results that we have seen. Although both tests have only been authorized for qualitative interpretation, the data for this research study have also been analyzed using the measured value. The cPass results are presented as % signal inhibition to reflect the relative amount of signal being blocked by neutralizing antibodies, while the specimen to cutoff ratio for the total antibody test has been normalized to represent a percentage of the maximum antibody level detectable by this assay. In donors previously vaccinated and not previously infected, all donors showed a strong immune response after the second dose of vaccine. Donor number 1 shows a progressive drop in response that may soon cross the 30% cutoff to become negative. Future time points for additional vaccinated, not previously infected donors will help provide further evidence suggestive of the duration of the immune response in this cohort. In donors previously infected with SARS-CoV-2 and not yet vaccinated, some donors show strong immune responses to both nucleocapsid and spike, while donor number 5 shows a stronger anti-nucleocapsid antibody response and possibly waning neutralizing antibody levels. Donor number 6 was actually negative for neutralizing antibody at all time points, despite having an immune response to the nucleocapsid protein, as evidenced by the total antibody positive results for all time points except the most recent time point, which was actually negative for total antibody. In donors who were previously infected, recovered, and then vaccinated, donor number 7 has maintained a strong immune response to both nucleocapsid and spike proteins since infection and post-vaccination. Donor number 8 was positive for both total antibody and neutralizing antibody after infection. An increased neutralizing antibody response was observed 29 days after the donor's first dose of vaccine prior to receiving the second dose, and this response has persisted beyond the second dose. The next case is particularly interesting and deserves its own slide. This donor was infected with SARS-CoV-2 and made a full recovery before starting on an immunosuppressant to treat another condition. This resulted in a rapid decrease in anti-nucleocapsid antibody levels, becoming negative at the third time point, while neutralizing antibody levels were a bit slower to wane. Following the first dose of vaccine, this donor showed a strong neutralizing antibody response, which has persisted beyond the second dose. We look forward to continuing to analyze specimens collected at future time points for the current cohort, while also expanding on the vaccinated cohort in hopes to better understand the immune response to SARS-CoV-2. One final real-world case study that recently developed is about a donor who gathered with a group of friends in January. The entire group tested positive following this gathering, except the donor who had mild symptoms of COVID-19, yet tested negative for the virus. 4 days after the donor had recovered, the donor began having urinary problems, which have persisted for 3 months. The donor asked, "Can your tests tell me whether I had COVID back in January?" And the donor added that he had gotten his first dose of vaccine a week and a half prior. We explained that a positive cPass test would indicate an immune response to the vaccination or infection, and if previously infected with SARS-CoV-2, we would also expect the Platelia total antibody test to be positive. We tested the donor's blood specimen and confirmed with a positive total antibody test that the donor had very likely been infected with SARS-CoV-2. Biodesix WorkSafe COVID-19 Testing Program has provided testing for key partners across the U.S., including Purdue University, the Big Ten Conference, Colorado State University, Chicago Public Schools, Major League Lacrosse, the State of Colorado, and Boulder Community Health, just to name a few. And we are excited for the opportunity to partner with GenScript to add cPass to our WorkSafe program. A test for neutralizing antibodies to SARS-CoV-2 complements the total antibody test, molecular diagnostic, and rapid antigen tests already offered. The research case studies presented today are reflective of other publications that have shown that vaccinated donors not previously infected with SARS-CoV-2 have a robust antibody response after vaccination. The response is specific for the spike protein after vaccination, but the strength and sustainability of the immune response is variable, especially in previously infected donors. Biodesix is dedicated to advancing lung disease research and clinical diagnostic testing. We look forward to expanding collaborations through both research and on-market studies in lung disease, as well as in helping to fight COVID-19 by exploring the duration of the antibody response after infection with SARS-CoV-2, exploring gene linkage by molecular testing for distinguishing replicating versus remnant nucleic acids, or leveraging our AI platform to identify those COVID-19 patients who are at higher risk of developing acute respiratory distress syndrome. If you are interested in learning more about how we could work together to fight the global COVID-19 pandemic, or on a lung disease testing strategy or other diagnostic testing related research study, please reach out. I'd like to thank you for your attention, and Sean and I will now address several of the questions that were submitted at registration, which we did not cover during our presentations. If you submitted a question in the chat during today's webinar, we will follow up with you individually in the coming days. I will now turn it back over to Elizabeth for the Q&A.
Thank you very much, Dr. Peak. And we will start with our questions. The first question is for Dr. Taylor. Will the cPass assay be able to detect neutralizing antibodies to current and future variants?
Can you hear me?
Yes, I can.
Okay, so the answer to that question is that we are actively producing the variant receptor binding domains. We have commercialized the UK and South African. I believe we're working on the key variants from India as well as in California as well. And these will be almost like plug and play into the cPass kit. So we actually currently have people that are using those variant RBD HRP complexes in the cPass assay. None of these have been EUA authorized, so this would all be in use with the research use only kits. The variants are coming faster than we can get them submitted to FDA. So we're working as quickly as we can to make sure people have access to these.
Perfect. Our next question, also for you, Dr. Taylor, can you speak to the neutralization capabilities of different subclasses of immunoglobulins, and why have you chosen to be immunoglobulin agnostic in designing cPass?
So it really wasn't a question of purposely being immunoglobulin agnostic. The fact is that we're using RBD as the bait protein for the neutralizing antibodies. And this assay specifically measures the interaction, of course, of RBD to the ACE2 receptor. So given that RBD is the key interactor with the ACE2 receptor, that molecule, that protein, can bind to any class of immunoglobulins. There's no way to make an RBD that would bind antibodies from one particular class of immunoglobulins versus another. There's speculation and there's some sporadic publications that are out there that claim that neutralizing antibodies are primarily generated from particular classes of immunoglobulins. I've seen one for IgA, but I am not confident that there's enough evidence that supports this type of conclusion. So my suspicion is that neutralizing antibodies are kind of like the dartboard analogy that I explained before. And it's more of a random type of a thing where the immune system encounters the vaccine and then just generates antibodies to all different parts of, for example, the spike protein. And in some individuals, it just doesn't happen that they generate neutralizing antibodies against RBD at all. And then in other individuals, they do generate the neutralizing antibodies to RBD and from whatever class of immunoglobulins those come from is also, I would suspect, to be fairly random. So whether it's IgG, IgA, or IgM, or a combination of all those. So the big advantage of this test is that it's able to measure the neutralizing antibodies that are generated, as opposed to just from one class of immunoglobulins.
All right. And this question for Dr. Peak: is the cPass kit able to be run on an automated platform?
Sure, Elizabeth, I can speak to what we've done here at Biodesix. So we've incorporated liquid handling robots in our laboratory to achieve high throughput testing with high precision and accuracy. We also utilize incubators, plate washers, and microplate readers to obtain a result. And we've developed a robust workflow to ensure high quality results are reported promptly to our customers. Sean, is there anything you'd like to add regarding automated platforms for cPass?
Sure. We have actively been working on this since we launched the cPass kit, and we actually have validated the assay on the TCAN Evo 200 system, as well as the Dynex Agility, and even the Biorad Evolus. Actually, we have a few labs globally that are using that system, and the kit actually works quite robustly on all those systems, although not exactly according to the EUA authorized protocol. But a lot of these systems that are walk-away automated ELISA systems, like the systems I mentioned, particularly the Dynex Agility and the Biorad Evolus, they only have single channel pipetting arms. So in order to accommodate that and the lags in time for pipetting on these systems, we've established a fully room temperature protocol for cPass. Again, it deviates from the EUA authorization, so it would have to be incorporated into a lab as a lab developed test for CLIA labs. But it works quite well, and it gives very, very similar data to, well, identical data actually to the pre-existing protocol that we have for the assay.
Thank you. And our final question that we have time for, this is for you, Dr. Peak. When will the cPass test be available?
Yeah, we plan to launch the cPass test as part of our WorkSafe COVID-19 testing program, as I'd mentioned, in the coming months. So if you're interested in developing a WorkSafe program with us or partnering with us for research or development purposes, please contact us and we can certainly discuss timelines to meet your specific needs.
Thank you so much. We have run out of time. I have compiled all of the questions that we did not get to, and I'll be forwarding those along to Dr. Taylor and Dr. Peak, and you'll receive those answers via email. I'd like to take this opportunity to thank our speakers, Dr. Laura Peak and Dr. Sean Taylor, and to thank the folks at Biodesix for sponsoring this webinar. Finally, I'd like to thank you all for coming and spending this time with us. We know life is crazy and difficult and demanding at this point, so we're very grateful you chose to spend this time with us. So on behalf of Cambridge Health Tech Institute's COVID-19 Breakout web series, I'd like to thank you all so very much and wish you a great day. Bye-bye.